Hello students. So we will come to the applications of animal biotechnology. See here in the applications there will be production of therapeutic proteins is one of the very big advantage of this particular animal biotechnology, wherein people are going to use animal cell lines. So they are going to prepare the stable permanent cell lines for particular means for the production of particular proteins. For example, like therapeutic molecules like insulin, somatostatin, there is molecules like growth hormones. So many other molecules are there which are therapeutically very important and commercially viable. These proteins are going to be, and these proteins are being produced by developing the specific type of animal cell lines. So specifically at the industry nowadays they are using the CHO cell lines, Chinese hamster ovary cell lines. So these are the cell lines which are being used for developing the stable transformants with the by transferring a specific gene into the cell line and making that particular cell line to produce the protein molecule as a secretory component, so that it will be available in the spent medium, which can be further purified by the downstream processing. So this is how people are actually producing these therapeutic molecules, and they are using it for the commercial purposes. So that part I'll give you details in the one more PPT recording. Now I'll talk about the transgenic animals, which is a very important application of the animal biotechnology, wherein in transgenic animals, what people are doing is they are developing the animals which are going to be having specific gene in it. So which can be an extra added gene, or it can be an enhancement of the production of a specific protein in the animal. And even people are also able to produce the knockout animals by using this particular technology, wherein they can make a particular animal unable to synthesize a specific type of protein molecules, or a specific genetic characters can be knocked out into that in that organism, so that that can be used for the study purpose. How people are then producing these animals? These are transgenic because they are going to have an extra gene or a transgene, nothing but it's a target gene is going to be transferred into this particular animal, as that gene is not going to be of the animal that particular species origin. So how they are developing these transgenic animals? One second. These transgenic animals are being produced, like for example transgenic maize, they are producing it by transferring the gene of interest by using a retrovirus vector, and that is being transferred into the organism through DNA microinjection method into an engineered embryonic stem cells, where they are going to use Cray-Logs-P system, and recombination system means Cray and Logs-P are going to be the promoter, and the genes which are going to be responsible for selection and which can be inducible, and through this they are developing the high capacity means they are using high capacity vectors for this particular purpose, and they are transferring into the organism, and thereby they are developing the transgenic maize. Application wise, these transgenic mice are being used for the study of different disease systems, for example Alzheimer's disease, and you as a test system in case of drug development, all these things. And even they are also used for studying the regulation of gene expression in case of conditional regulation, control of cell death also. They are also doing this particular methodology is being used for cloning in livestock by nuclear transfer method. They are developing transgenic sheep, cattle, goat, pigs, birds, even fishes also. So how this is being done? They are using the retroviral vector systems for the transfer of the genes. So how they are doing it is that, for example, you can see they are going to take a donor female mice. From there they are develop means the embryo is going to be taken out, which is going to be of approximately 8 cell stage. Now this particular embryo is going to be subjected for transfer of the gene by the process of microinjectile method, either through microinjectile bombardment method or by using microinjection. So they are going to transfer the retroviral gene construct, the recombinant DNA gene construct into this particular what you call the embryo. Now once it is going to be transferred into this embryo, now this particular embryo is going to be stabilized in the in vitro, and then again it will be transferred back into the female. So now once this female is implanted with this embryo, the embryo is going to give rise to the progenies, and these progenies, one of the at least the progeny is going to carry our transgene, so which can be selected based on the marker present within that particular transgene, which is going to develop a new characteristic in that particular mice in terms of producing a specific protein. So this is how the transgenic mice is being developed. So here the transfer of that particular gene into the organism, it is also people are doing it in another way, wherein instead of taking an 8 cell embryo, they can take a fertilized zygote that can be taken, and in that particular zygote they are introducing the transgene into the male pronucleus by the process of microinjection. So wherein the fertilized zygote is going to be, or egg is going to be holded with the suction through a micropipette, and from another side by using a small injectable syringe. So they are going to transfer the transgene into the male pronucleus and allow the fertilized egg, then it will be implanted into the surrogate mother, and once that particular mother is going to develop the progeny, the mice are going to get delivered. So these mice, any one of them are going to carry our gene of interest. So here we can check that particular existence of our DNA or gene of interest in that particular mice by using the screening methods, where nucleotide hybridization methods are going to be used through the using an RNA, where they are going to convert the RNA into cDNA, and that cDNA can be sequenced or can be blotted in order to identify the presence of the target gene in that particular DNA of that organism. So thereby they can able to screen and identify which particular progeny is carrying the gene of interest, and it can be further used for the study. So this is a microinjectile method only into the not into the embryo, here it is into the fertilized egg. So here you can see it is little bit different one. So in this case in vitro fertilization only will be done, not taking the fertilized egg from the female, but instead they are going to take the egg is going to be collected from the female, they are going to collect the sperm from the male, and then they are going to do in vitro fertilization. This in vitro fertilized egg can be further used for microinjection. This is another method. So there is one more method wherein they are going to develop the genetically engineered embryonic stem cells. For that, instead of taking a fertilized egg, they are going to take a blastocyst from that particular mother, and cells from the blastocyst are going to be subjected for microinjectile bombardment method, and the cell which is carrying the gene of interest, the transgene or the recombinant DNA construct, is going to be selected by the process of enrichment, and the transfected cell, once it is selected, it will be allowed to multiply, and the multiplied transfected engineered cell will be again added back into the blastocyst through the microinjection method. Then that particular blastocyst carrying the embryonic stem cells will be implanted into the surrogate mother. So then the progenies are going to be screened for the presence of the transgene. So this is one more way people are going for genetic engineering and developing the transgenic animals. Okay. So this is also people are going to be doing in another way. You can see that. So that is establishing the transgenic animals using engineered embryonic stem cells. The same thing which I told you just now. So this is what from the male and female they can collect the samples, then induce the fertilization, then develop the blastocyst. From that blastocyst they can take the cells into the petri plate, and these particular cells are going to be subjected for centrifugation and remove the inner cell mass is going to be removed, and the inner cell mass is going to be dissociated with the trypsin. That trypsinated cells are going to be allowed to grow in a petri plate, forming a feeder layer. From there it can be used to develop the that can be further induced to develop into a particular lineage, and the cell can be subjected for through the subjecting for differentiation, and these differentiated cells further can be developed for transferring the gene of interest, or in other way these particular embryonic stem cells can be used for the that can be transferred with the transgene, and then the transgene having embryonic cell can be enriched and it can be transferred into the blastocyst and implanted into the mother. Or we can also develop that particular feeder layers, and then that can be multiplied for selections, and these particular feeder layers embryonic stem cells, the transgenic embryonic stems carrying the transgene embryonic stem cells, that can be that screened and selected cells can be multiplied and can be kept for further utilization and studies. So this is one other way. So these engineered transgenic animals they can be done, as I told you, embryonic stem cells. So this is the same methods, genetic engineered cells, place them into the back into the the cells are transferred into blastocyst and then back into the animal, which is going to produce the progeny carrying the target gene. So these transgenic mice, why they are doing it? To produce high capacity vectors are going to be used here for the transfer of the gene. Because here, why high capacity vectors? Because the vectors should able to carry a large gene, and it can be microinjected into that particular animal. So a number of examples are there which are being used, means number of animals people have developed for this. There are some transgenic models people have developed, model animals, where for Alzheimer's, amyotrophic lateral sclerosis, Huntington's, arthritis, dystrophies, tumorigenesis, hypertension, neurodegenerative disorders. Many models have been developed, model systems have been developed. These transgenic animals are also used as a test system. For example, in case of CFTR, they have made the production of this particular protein into the milk of the animal. They have tested it, and also for protection against the metastasis, which is caused by Staphylococcus aureus. So they have developed these kind of testing systems, and even for conditional regulation, for example, tetracycline inducible systems have been developed. Conditional control of cell death, they have developed the models for studying the organ failures, like this. So they have produced a number of human proteins into the animal systems, as I was talking about the therapeutic proteins which have been developed with the cells and as well as transgenic animals, and made them to secrete into specific component of these animals, which is further used for isolated and used for therapeutic purposes. So I'll stop here. I'll continue with the another set of recording later. Thank you.